All right. Good afternoon, everyone. My name is Rodolphe Clerval. I'm the CEO of Coave Therapeutics. We are a gene therapy company focused on chronic retinal diseases. Okay, so we are developing basically gene therapy for wet AMD and GA, and for gene therapy to succeed and find their place in the therapeutic arsenal, those gene therapy should be in-office administered with a very low burden, should have a safety which is comparable to current biologics and standard of care, and will enable an ultimate durability. So gene therapy is a modality that could deliver the ultimate durability. But when we look at the current first generation gene therapy, for instance, for wet AMD that are in late clinical stage, those products have demonstrated that they can improve dramatically durability. There's no question. There's no question those products will go to the BLA and make it, but they have not fulfilled completely this key element. First, the IVT gene therapy required 5 to 7 months of steroid prophylaxis, which is a major burden for the patient and for the physicians. And when we look a little bit more in detail about the efficacy they have proven, despite reducing dramatically the number of biologics injections, more than half of the patients still need rescue biologics after 1 year. Those IVT gene therapy have a PK issue. The companies have provided a lot of PK data through the phase 1-2, and when we look at the trough concentration of aflibercept in the aqueous humor, we see that the level of aflibercept they achieved is always below the trough concentration of aflibercept after 4 weeks. And we see a lot of variability. And actually, among the patients, despite durability of production of aflibercept, there's half of those patients that achieve a suboptimal level of aflibercept in situ and require then an additional biologics administration. And why is that? It is because of the delivery. It is because of the route of delivery and the vector they are using. The intravitreal AAV vector that have been developed, they are actually have a poor transductions of the posterior segment and a massive transductions of the anterior segment of the eye. And the consequence of that is inflammations and inflammation that require this steroid prophylaxis. As opposed to that, actually REGENXBIO, using the suprachoroidal route of administrations, have demonstrated that it's a much safer route that for most of the patients doesn't require the use of steroid prophylaxis, despite using actually a dose which is tenfold the dose used by IVT product. Unfortunately, they use AV8, which was not a serotype designed on purpose for this route, and AV8 has a very limited transductions of RP on the back of the eye. So Coave, we are actually, the company is built around the delivery technology on new platform. So we have developed new vectors that have been developed specifically for the suprachoroidal route, and these vectors have the same safety or even better safety profile than AV8, but has a very much higher potency to transduce RP and photoreceptors. And we have actually conducted to date 4 NHP studies that has evaluated our capsid versus the benchmark, AV8, 7M8, and others. This is the third study here. You can see that we compare our vector, the Coave SCS, at the bottom of this slide, with the AV8. And what you see first is there's a much broader transductions of the retina, the white spot here in these pictures. And when we look more at the level of the cell that have been transduced, RP and photoreceptors have been massively transduced in the periphery and the mid-periphery of the retina. We have a fourth actually studies which is ongoing and will deliver very soon with aflibercept as a transgene. We are using this capsid, the supracaudal route, that we combine with actually a payload. This payload will replicate the mechanism of action of faricimab. So it's a combination of an anti-VEGF protein and anti-Ang2. So we are actually building the best of what we can do in gene therapy, the best route of administrations, capsid, and payload. We are currently in IND-enabling study, where we will enter in clinical next year through two track of development. One is an IIT in China, where the first patient is anticipated mid-27, and the second route of developments will conduct in parallel for an IND and CTA. So this is Coave. We have raised last year a 32 million euro round with Invus and Novo as the lead investors. We're developing this program, and we're developing also a second program using the same capsid and the same route of administration. Thank you.